Excuse me, do you know what the internet key is for APS guest?
It's Franklin, capital F,
Yeah.
1744. Yeah, those are the
Is Jeremy ready? Good afternoon. Friends, we can begin. I'm Roger Bagnall. I have the privilege of being the president of the American Philosophical Society, and it's my great pleasure to welcome you to Philadelphia and to the spring general meeting of our members. We have about 300 people registered for the meeting this week, which is kind of close to a record, I think. Thank you all for joining us. We also have 23 members attending their first APS meeting, who will be formally inducted into the Society over the next 3 days. The new members are wearing name tags that have red ribbons on them, so please welcome them to the Society when you see them. Now, a few housekeeping items. I'll bet you could all stand up and in unison recite the first one, which is please turn off your cell phones. Second, we do have a relatively full house, so please make sure that all seats are available, as they say on the airlines. One other thing, there is a selection of items from the APS's library and museum in the display cases on that side of the stage and this side. These are inspired by themes related to today's meeting talks. On the left, and I'm not sure, is that stage left or, yes, are items related to bioinspired technology from the 18th century botanical printed currency, yes, that's over there, to a 20th century device developed to study enzymatic relations. On my left, your right, are items from the collection of Beatrice Mintz, an American embryologist and APS member whose work on genetic modification led to important advancements in cancer research. Now we begin the formal meeting, as always, with the necrology. I'll ask you to stand while I read the list of members who have died since we last met. Claude Allègre on January 4th, 2025, at age 87, elected in 1992. Floyd Bloom on January 8th, 2025, at age 88, elected in 1989. Michael Boudin on March 24th, 2025, at age 85, elected in 2010. Victor Brombert on November 26th, 2024, at age 101, elected in 1987. Jimmy Carter on December 29th, 2024, at age 100, elected in 1991. Joanne Chory on November 12th, 2024, at age 69, elected in 2015. Lauren Graham on December 15th, 2024, at age 91, elected in 1995. Robert Hazelkorn on February 10th, 2025, at age 90, elected in 2014. Christopher Jenks on February 8th, 2025, at age 89, elected in 2004. Charles Longsworth on January 8th, 2025, at age 95, elected in 1990. Martin Marty on February 25th, 2025, at age 97, elected in 1994. Jeremiah Ostreicher on April 6th, 2025, at age 97, elected in 1994. Colin Renfrew on February 24th, 2024, at age 87, elected in 2006. Stuart Rice on December 22nd, 2024, at age 92, elected in 1986. Keith Thompson on February 21st, 2025, at age 87, elected in 2011. Lartzert Sif on November 25th, 2024, at age 97, elected in 2006. Thank you. We now proceed to the induction of new members.
Will Dr. Francis S. Collins please come forward to the President's desk to sign the roll book, and then receive a formal welcome from the President? Mr. President, I have the honor to present Francis S. Collins, former Director of the National Institutes of Health. As the longest-serving director of the NIH, spanning 12 years and 3 presidencies, Dr. Collins oversaw the work of the largest biomedical research institute in the world. As a physician geneticist, his career has been dedicated to human genetics, serving as the director of the National Human Genome Research Institute of the NIH for 15 years. He led the International Human Genome Project, which culminated in 2003 with the completion of a finished sequence of the human DNA. Dr. Collins is the recipient of the Presidential Medal of Freedom and the National Medal of Science for his work in genetics.
Francis. By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Stand here for the name.
Congratulations. I'll get on the other side.
Between us. Thank you.
Will Dr. Bernard L. Faringa please come forward to the President's desk to sign the roll book, and then receive a formal welcome from the President? Mr. President, I have the honor to present Ben Faringa, Jacobus H. Van Hoff Distinguished Professor of Molecular Science at the University of Groningen. His career began as a research scientist at Shell in the Netherlands and at the Shell Biosciences Center in the UK, before he was appointed lecturer and then full professor at the University of Groningen. As a synthetic organic chemist, Dr. Faringa has contributed to the development of molecular machines, applying the idea that components of the smallest machines could be molecules, and that to function, the parts of a machine must move relative to one another. In 2006, he received the Nobel Prize in Chemistry for the design and synthesis of molecular machines.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Over here for me.
Will Dr. John Dupre please come forward to the President's desk to sign the roll book, and then receive a formal welcome from the President? Mr. President, I have the honor to present John Dupre, a professor of philosophy of science at the University of Exeter and consulting director of EGENIS, the Center for the Study of Life Sciences. Dr. Dupre founded EGENIS in 2002 and was the founding director for 20 years. Under his leadership, the center earned a reputation for its innovative, interdisciplinary approach to the ethical, social, political, and philosophical implications of life sciences. Dr. Dupre's research largely focused on the philosophy of biology and metaphysics. His most recent work has explored a process metaphysics grounded in contemporary biology that examines the implications of process philosophy to questions of personal identity, human nature, humankind's, and free will.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Just come over here for the program. Got it.
Will Dr. Kaelin Carrico please come forward to the President's desk to sign the roll book, and then receive a formal welcome from the President? Mr. President, I have the honor to present Kaelin Carrico, professor at the University of Szeged, Hungary, adjunct professor of neurosurgery at the Perelman School of Medicine at the University of Pennsylvania, and the former senior vice president of BioNTech. Dr. Carrico's research focuses on RNA-mediated mechanisms with the goal of developing in vitro transcribed mRNA for protein therapy. In 2005, she and her research partner, Drew Weissman, discovered that certain modifications of the building blocks of RNA prevented unwanted inflammatory reactions and increased the production of desired proteins. Dr. Carrico's discovery laid the foundations for the mRNA COVID-19 vaccines developed by Pfizer and Moderna to fight the global pandemic. Along with Dr. Weissman, Dr. Carrico was awarded the Nobel Prize in Physiology or Medicine in 2023 with her contributions to the COVID vaccine.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Just come over here for a photo. Photo.
Thank you. Will Dr. Michèle Lamont please come forward to the President's desk to sign the roll book and then receive a formal welcome from the President? Mr. President, I have the honor to present Michèle Lamont, Robert I. Goldman Professor of European Studies and Professor of Sociology and African American Studies at Harvard University, a cultural and comparative sociologist who studies morality, group boundaries, and inequality. Her research ranges from such topics as dignity, respect, stigma, and racism to methods of evaluating social value. Dr. Lamont served on the faculty of the University of Texas at Austin and Princeton University before joining Harvard in 2003, and was the 108th president of the American Sociological Association. Dr. Lamont's essay, Prisms of Inequality, Moral Boundaries, Exclusion, and Academic Evaluation, won the 2007 Erasmus Prize for her contributions to the relationship between knowledge, power, and diversity.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Patrick? Patrick. Got it. Thank you.
Will Richard C. Levin please come forward to the President's desk to sign the roll book and then receive a formal welcome from the President? Mr. President, I have the honor to present Richard Levin, President Emeritus, Frederick William Beinecke Professor Emeritus of Economics, Professor Emeritus of the School of Management, and Director of the Project on U.S.-China Relations at Yale University. During his time as President of Yale, he invested over $5 billion in the university's facilities, supported economic growth and home ownership in New Haven, set a goal to reduce campus greenhouse gas emissions by 43%, established Yale's Office of Sustainability, strengthened the university's science, engineering, and medicine programs, and began extending need-based financial aid to international students. It's quite a lot. In addition to his academic appointments, Dr. Levin served on President Obama's Council of Advisors for Science and Technology, was the CEO of the online learning platform Coursera, and served as director of American Express.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Let's come over here for photo. Got it.
All right. Will Dr. Jonathan Losos please come forward to the President's desk to sign the roll book and then receive a formal welcome from the President? Mr. President, I have the honor to present Jonathan Losos, Distinguished University Professor, Professor of Biology, and inaugural Director of the Living Earth Collaborative at the University in St. Louis, Washington University in St. Louis. An evolutionary biologist and herpetologist, Dr. Losos researches the behavioral evolutionary ecology of lizards and the evolutionary diversification of lizard clades. As director of the Living Earth Collaborative, which facilitates interdisciplinary research among plant and animal biologists in partnership with Washington University, the Missouri Botanical Garden, and the St. Louis Zoo, Dr. Losos leads the Center's scholarship on biodiversity and sustainability.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations.
Will Dr. Kathleen McEwen please come forward to the President's desk and sign the roll book, and then receive a formal welcome from the President. Mr. President, I have the honor to present Kathleen McEwen, Henry and Gertrude Rothschild Professor of Computer Science at Columbia University. She served as the founding director of the Data Science Institute and as Vice Dean for Research for the School of Engineering and Applied Science at Columbia. She was the first woman tenured in Columbia's engineering school, the first female computer sciences chair, and founded Columbia's Women in Computer Science student group. Dr. McEwen's research focuses on natural language processing, summarization, as well as natural language generation and analysis of social media. She strives to positively impact society through projects such as the automatic generation of updates on disaster and the analysis of emotion in social media to identify signs of grief in response to current events.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Over here. Photo.
Can you get me a napkin? All right. Will Professor Dr. Herman Partzinger please come forward to the President's desk to sign the roll book and then receive a formal welcome from the President. Mr. President, I have the honor to present Herman Partzinger, President of the Prussian Cultural Heritage Foundation and the Executive President of Europa Nostra. In his time at SPK, Professor Dr. Partzinger has led the renovation of Museum Island and the realization of the Humboldt Forum in the reconstructed Berlin, Paris. As a historian of prehistoric archaeology and history in Europe, the Near East, and Central Asia, he has led archaeological projects in Spain, Turkey, Serbia, Mongolia, Kazakhstan, Uzbekistan, Tajikistan, and Iran. Probably some other places too. His research includes archaeology, history, cultural heritage, and cultural and academic policy issues relating to early sedentary life and the domestication of plants and animals.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Photo. Yep.
Great. Okay. Will Dr. Cecilia E. Rouse please come forward to the President's desk to sign the roll book and then receive a formal welcome from the President. Mr. President, I have the honor to present Cecilia Rouse, President of the Brookings Institution, Kotzman Ernst Professor in Economics and Education, and Professor of Economics and Public Affairs at Princeton University. She served as the chair of the Council of Economic Advisors during the Biden-Harris administration, the first Black American to hold the role in the CAA's 75-year history. She was a member of Barack Obama's Council of Economic Advisors and worked on the National Economic Council as a special assistant to the president under Bill Clinton. As a labor economist with a focus on the economics of education, Rouse is the founding director of the Princeton Education Research Section, which strives to discover educational policies and practices that make educational opportunity universal, prepare students as future stewards of education, and support ongoing educational improvements.
By the authority and in the name of the American Philosophical Society, held at Philadelphia for promoting useful knowledge, I do admit you a member thereof. Congratulations. Come over here. Come over here. Yep. Good. Okay.
I think we're good.
All right. It is now my pleasure to hand over the chair, or the lectern as it may be, to Vice President Jacqueline Barton to introduce our afternoon speakers. Jackie.
Thank you, Roger, and welcome everyone to this first session. I'm Jackie Barton. I'm a chemist from Caltech, and I'm going to be chairing this session. It's my pleasure to begin by introducing Joanna Aizenberg. Joanna is the Amy Smith Berylson Professor of Materials Science, Professor of Chemistry and Chemical Biology, and Director of the Kavli Institute for Bionanoscience and Technology at Harvard. She received her bachelor's from Moscow State University and a PhD from the Weizmann Institute. After a postdoctoral stint at Harvard, she was a member of the technical staff at Bell Laboratories for several years before returning to Harvard. Aizenberg is very much a pioneer in the chemistry of bioinspired materials, and I believe there are some over there that you can look at after the session. What Joanna does is she studies the basic chemical principles that underpin biological architectures, and then using these principles, she develops new advanced materials and devices. Her work offers broad applications in architecture, medicine, energy, even robotics. And as you will see, this is extremely creative work and quintessentially Aizenberg chemistry. Among her numerous awards are the MRS Medal from the Materials Research Society, the ACS National Award in Colloid Chemistry, and the ACS Ronald Breslow Award in Biomimetic Chemistry. She's been elected to the National Academy of Sciences, the National Academy of Engineering, the American Academy, and was elected to the APS in 2016. Her talk today is titled New Bioinspired Materials When Biology Meets Chemistry, Physics, Engineering, and Design. Please join me in welcoming Joanna.
Thank you, Jackie. It's a very kind introduction. What I will try to do is to show that many sciences these days should come together and create new knowledge, and this society is about new knowledge, because so many things we can do not only within the boundaries of your own research, but when we exchange our sciences, our knowledge, and bring together biology, material science. I'll give you some examples. We'll see how many I will be able to present, and to think how that can change life and make new, better materials, better structures, better architectures. And to begin, let's say we want to dream. And if we're dreaming and we think about imagining new technology, so what would it be that we want to have in our life? Let's say it would be materials, devices, architectures. You can help me even to build this list. We want them to adapt to their environment. We want them to harvest energy. We want them to self-heal. We want them to reconfigure maybe in some more useful organizations. We want them to collect and deliver water, self-clean, whatnot. There is so many things that one can put on this list. What I will try to convey here, that at least I feel that a lot can be learned if we think about it as a vision, building as organism. And when I say that, I would say that it's very easy to think about the first part of this list, which is to search for biological systems with interesting properties. Anything in biology is interesting. More difficult is to understand what are the mechanisms of their formation. What is it that we can learn from these biological systems to use to our advantage? It doesn't have to be as sophisticated as biological systems. I don't think we're there yet. But then, hopefully, by using this multidisciplinary approach, we can apply it to novel materials, novel medicine, and so many different things. Since there's very limited time, I will today limit it a little bit, although you will see there's a lot that I want to put in this talk. And I will limit it to somewhat plant-inspired materials. I'll talk about lotus leaf, pitcher plant, Venus flytrap, iridescent seeds. We'll see how far we will go in this list, just to show the breadth of sometimes strange ideas that can come up, that we can come up with if we think about biology as a driver for new technologies. So, chapter 1, lotus leaf. Now, we all know how clean and wonderful the leaf is, and the reason is that it's what is called superhydrophobic. It has a structure that hates water. Superhydrophobicity comes from the fact that it hates water, but it is structured. So water is sitting on a cushion of air, and therefore is highly mobile, and when it is picked up, and rolling from the surface of the lotus leaf, it picks up dirt, and therefore lotus leaf is always clean. This phenomenon is called superhydrophobicity, and just to show you what is behind it, let us say we have hydrophilic substrate. It likes water, water spreads. If you have hydrophobic, it doesn't like water, but it's still in contact with it. If now you have this superhydrophobic surface, water cannot even stand on it. It jumps off, leaving surface completely free of water. It's an excellent approach to think about self-cleaning materials, but the problem, and I can already say, chapter 1 ends here. I'm already moving to another biological inspiration, because nature evolved it to deal with water, not to deal with really complex environments, with biological media, with icing conditions, in high humidity. Pretty much anything but not water would get you from these super non-wetting condition to super wetting condition, when the liquid, contaminated liquid, when the surface is damaged, will actually get in. The example that I show here, and I will come back to this example later again, this is morph butterfly. The color of this beautiful blue color is not pigment. It is what is called structural color. It's based on a structure. I'll come back to this later, but what I want to show here, it was optimized to be superhydrophobic. Water jumps off, color remains, and actually scales remain intact, but if it's different medium, in this case it's alcohol, it actually penetrates into the structure, becomes this super sticky condition. In this case, it's manifested by change of color, because color is now removed. So it doesn't work. And if we are to think about other sticky problems of our life, and I know that it's very difficult to read what is here, but intentionally done so, what says here is that materials get dirty. They do. And most difficult things, if we think about snow that we don't want to be collected on our cars, as you could see here, or if we want to clean our water, this contamination is incredibly difficult to get rid of. I can again make a much longer list if we think about infections. Again, sticky substrates that we want to avoid, or blood clots, and going to marine fouling. Our solar batteries, they cry, please clean me, because you develop all these technologies to work well, but if it's dirty, there is no way for them to work the way we expect. And graffiti, I'll talk about that as well. I really don't like graffiti. So we can change many things if we prevent material structures from contamination, and that will affect energy, health, environment. So what is the better strategy that we can use? And I will show you one. And in this case, it's chapter 2. It's pitcher plant. Here's pitcher plant. That is really interesting. It likes to eat these nice creatures. But suddenly, on a wet day, they cannot run anymore. They actually sliding into the digestive juices of a pitcher plant on their own. It doesn't even have to capture them. So something interesting is happening here when insects cannot run. On the dry days, everything is fine. The moment it's wet day, it's high appetite. They're running into the mouth of that plant. So what is happening here? Let's just, very short, I would not give you a very detailed description of that. Interestingly, the structure of this plant is very similar to the one of the lotus leaf. It's also structured surface. So there is a structure there, but it's now not hydrophobic, it's hydrophilic. When you have structured hydrophilic surface, it loves water. So what it does, it picks up, on a wet day, it picks up a layer of water that now covers the structured surface, and it becomes water that is seen by the oily insect feet. The outcome of that, oil-water interface, is very slippery. Everything slides. In fact, it doesn't have to be pitcher plant. Same principle is in our stomach, in the scales of fish that allow them to swim better, to protect our gastric lumen or our eyes, and this is eyes of my grandson, but there is always a layer of liquid. Let's think about these structured surfaces with a layer of liquid on them. And based on that, my lab developed what we called slippery liquid-infused porous surfaces, or SLIPs. And they're really omniphobic. They hate everything. Nothing can stick to these surfaces. The principle is kind of counterintuitive. We're using roughness to create non-rough, ultra-smooth surfaces. What happens there is that in roughened surfaces, if it's properly chemically functionalized, you now have a way to capture a lubricant. It doesn't have to be water, as it is in pitcher plant. It could be any lubricant that I can choose, and that keeps this lubricant within this porous network, which means it creates a smooth, slippery interface from which everything slides, and in this way, one can say, slippery surface. Everything slips. Let me show you a couple of examples of what is happening on these surfaces. This is PBS Nova. As I said, I don't like graffiti. That was in my lab. What is talking about that there is no graffiti on the slippery side of things. I can show many more examples. Let's remember, and it's very important, that since its liquid is now active part of the material, it means that it has self-healing capabilities. So superhydrophobic surface, crude oil is not moving because it's really difficult medium to handle, but self-healing takes place on a slippery surface. I just shown a couple of examples. It doesn't have to be exhaustive. Here is an example of how it deals with anti-icing. The same substrate, the same cold plate. There is ice, frost actually, forming on one side, but there is no ice or frost forming on a slippery surface. Or if we think about biocontamination, this example that I show, first of all, shows that we can make them, if we want, transparent. So as half of the glass is just regular glass, and the other half is made slippery glass. And you could see this algae layer is not forming, well, it's forming, but not attaching to the glass slides. So what can we do with that? A lot. For example, if we think about medical devices, I'll just show one potential application in bronchoscopy. You need your lenses to stay clean, even if they're in contact with biological medium. For example, in this case, I show the case of biopsy performed on pigs with the slippery surface. You dip it in blood, but it immediately shows the image. So there is no contamination. You continue the biopsy procedure and endoscopy imaging. Let's think about applications in marine fouling. So here's a couple of examples of testing these materials in the form of paints compared to regular materials that are used now, you could see that slippery surfaces, as a paint for marine application, can keep the surfaces clean of fouling. And I will show just another one. So going from just demonstrations to actual applications, the one on the right is showing the application in water harvesting. And what you should see here, that this side is not just slippery surface, it's a little bit more. And I will mention now the merger between different approaches. In this case, just slippery surface will not be sufficient, but if we combine the lessons from from other animals, in particular from desert beetle that develops very interesting bumps on its surface, but also from cactus that has asymmetry in its needles that allows water to be transported in a specific direction, we can design the surfaces that can collect water. And I can tell you that while this one is nothing is even happening, the amount of water that can be collected on the surface using these three, combination of these three approaches is about 2 orders of magnitude than what can be done if we're talking about water harvesting on regular materials. Okay, chapter 2 is done. Chapter 3. Yet another interesting case. I will talk about wheat-ons, and you probably know, if not, I will tell you that they're really interesting ability to to introduce themselves into ground deeper and deeper, simply by changing an angle between the ons that change the force that they have on putting them into ground. So they seed themselves better and better. Just opening flowers would be another example to mention. If we think about Venus flytrap, the common feature, and if cilia and flagella has a lot in common with that, or at least the way I want to simplify these very different biological lessons to maybe a set of simplified rules that would be sufficient for me to create reconfigurable dynamic materials. So if I were to ask, what is the common feature, simplest common feature here, it is reconfigurable, moving, high aspect ratio, hairy materials that are put in motion by a muscle. So what can we do as material scientists or chemists to do that? What we can create is what we call hairs, which is hydrogel actuated integrated responsive structures. So what we actually do, we create these high aspect ratio hairy surfaces, but we place a layer of gel that acts a function of a muscle, that when the gel, as it responds to external stimulus, contracts, the fibers go down. When it expands, they stand up. So what happens then, we pretty much using gel for changing our hairstyle. Let's see what we can actually do with this approach. As chemists, as material scientists, we can do many things. We can either change the hair, we can use different products on our hair, we can make them hydrophobic, hydrophilic, put something on their tips, or we can change the styling gel. And there are muscles that we can develop that respond to pH, to temperature, to different chemicals, to light. And that would put the materials in motion. And the outcome of that is just, let's take a look how these high aspect ratio materials put in motion. They stand up, they lie down. But when we want to use them, so what can we do with these structures? This example tells you that we can create fabrics that are super hydrophobic when they're standing up, which means in the rain, nothing gets wet. But on a dry day, when you want moisture to actually to go through your the materials that you wear, it becomes switchably hydrophobic or hydrophilic. You can actually use them, and we do use them, as a way to change our windows, that depending on the temperature outside, it will either let the light through or stand up and change the amount of light coming through, or simply create materials that change color or pattern in response to the stimulus. Yet another example shown here, you can use it to hide messages, because only when they respond and bend, certain messages become readable, and they're not such when they're standing up and away from each other to interact. Okay. 2 more slides, maybe. I already see the time flies. Chapter 4, I want to talk about tulips, but specifically this Queen of the Night tulip. These iridescent seeds of Margaritaria nobilis, this blue color is not a dye. It is a structural color, such the same way as Morpho butterfly has it. In other words, and we probably all know that opals don't have a dye in it at all. And the structure of opal, it's composed of glass. It's S-iotospheres that are, depending on their size and depending how they pack, they form an array, a photonic crystal, that the color will depend on this packing. So that is the case for the ones that I've shown here. And this is just to show you another butterfly, Sesotris butterfly. This green, beautiful, beautiful green patch is structural color. It's actually what is called inverse opal structure. So where there were spheres, now empty spaces, we can make them. We can make them and have this structural color also made out of glass in this case. But we remember that butterfly, if it's not water, then it can infiltrate the structure. It's not used by butterfly for interesting applications or interesting functions. But I want to show you another organism. I've done a lot of work on this one. This is a relative of a star fish, and this is what is called brittle star, that actually changes color during the day and during the night. It was supposed to be, at least in the literature when I started to work on it, said that it's a biomimicry approach. No, it's not mimicry, because then you would expect that this creature would be white during the day and black during the night, so that to avoid predators. So it's exactly opposite of biomimicry approach, and what appeared to be the case is that, in fact, it has porous network that when pigment comes and covers the lenses during the day and withdrawn back during the night. It's active optical system where liquid changes optical properties. In fact, what is it doing? Just really is sunglasses that change to be sunglasses or not to, depending on availability of light. So this is interesting, but can we now combine the lessons from these two organisms and think about dynamic optical devices? What if we use different chemistries in our colloidal crystal and functionalize them with different molecules that be more or less hydrophobic or liquid phobic? And therefore, I can encrypt so any messages I want. Here's one nice demonstration. I've done a lot of work with marine creatures. Where liquid comes in, the structure is filled, the color is gone. So you can have a whole range of materials. This is, by the way, mine was coming from Air Force, and this is butterfly that we've done it to it, and it says U.S. Air Force, and then it goes back when the liquid is evaporated. So what you can do, you can have rapid identification of of liquids. You can use it for medical sensing. In particular, we've shown how to apply it in diagnosis of neonatal jaundice. There are many other things that you can do, or you can just do it for fun. And in particular, we had design students using it and showing how that can be used in your bathroom. When you take a shower, a lot of nice patterns appear, but the moment it dries out, it all disappears, and it really nice tiles in your bathroom that respond to environment because they're now sprayed with water, and only the places where water cannot come in remain colored. Here's a Harvard sign with, and this is a wonderful project when students decided to have a chess play. That color depends on the size of your pores, but to know exactly which piece this is, you have to put a specific liquid in it, and you need to know which one is that. So, I will finish with showing last example, and this would be deep sea sponge that is called Venus's flower basket. Has nothing to do with flowers, but I really wanted to show this example as well. Its name is Venus's flower basket, but this is actually deep sea sponge that lives deep in the ocean, and it has in its structure one of the best optical fibers that we know, that are pretty much better than many of optical fibers that we make, but it's made of glass. It's one of the strongest glasses I know. In particular, I was even able to teach almost entire class in mechanical engineering based on the principles that this sponge is using in designing of its skeleton. And what is interesting, that in fact it is an illuminated glass house of the deep, because not only it resembles, and it's really strong material that resembles architectures that we use now. It's about a different lens scale a little bit. But it also has a fiber optical network in it. And in fact, it is used by a pair of shrimp that lives inside the sponge, that takes advantage of the optical network that is connected to the ocean floor, taking light from the bioluminescent bacteria. In this way, it is a shining beacon deep in the sea, so food is attracted to it. And deep in the ocean, it creates really optimized materials for strength, fiber optics, and so on. So I will not talk about this. I just want to finish with talking about these principles. I want to say, let's all, especially in these difficult times, let's go and smell the flowers. I can, in addition to that, discuss with you which principles of smelling we need to take into account in our research if we design chemical sensors. But instead, I will summarize saying, let's do it together. We do need to bring together different sciences, different approaches to understand and principles which we can combine and create new materials. And let's not forget, we do not have to mimic one organism at a time. A lot of interesting strategies can come up by bringing together sometimes orthogonal strategies that are developed by different organisms, and in this way we can design really interesting materials. At the end, I want to thank my group. It's really eclectic group from every science possible in my students, and I want to finish with that. I hope that with our approaches, we can keep it clean and not contaminated.
Well done. Thank you. Well, I know Dr. Aizenberg would enjoy answering questions. They should be short, and please say your name and where you're from. Thank you. There's one here in the back.
Rich Schifrin, Indiana University. That was a great talk. You gave wonderful examples of biology helping you design materials. How about materials helping you design biology?
I hope that can be done as well. Probably not in my lab, but we do use, in fact, I've done in my PhD, I've done a lot of work in using different biological molecules to change the properties of crystals and so on. So I think both are entitled to help each other, and I'm not really sure where there would be more help, coming from biology to help material science and engineering, or the other way around. I think both fields, or many other fields, can benefit from this two-way street.
Other questions?
Meadows, Philadelphia. I never knew so much about the structure and function of materials, but I'm wondering, these changes that come about that are functional and structural, they're controlled by genes, aren't they? They're genetic changes.
That's true.
Is anybody looking at the genes that make these changes possible?
Absolutely. In most cases, in most interesting, especially characteristic systems that are studied in biology, we are looking what is genetic makeup, which proteins, what is from the biology side that drives these materials, how they evolve and what is happening to them. What I was trying to say, that we can reduce the complexity to a manageable set of principles that are not exactly the same as biology. That's why I would rather call it bioinspired than biomimetic, not mimicking nature by taking lessons and reformulating them to use different materials, and instead of genes, to have other ways of encoding information. And we have the ways to encode information. We also have the ability to use different materials. Nature is actually quite limited in the repertoire of materials it has. We can use steel for something that doesn't exist in nature, but it brings yet another level that we can use. But key feature is to extract manageable set of lessons that can be reformulated to the function or to the type of application that you have in mind.
All right, I think with that we'll go on, and let's thank Joanna once again for a beautiful talk. Our second speaker today is one of the newest members of the APS, William Kaelin Jr., who was elected in 2024 and who will be formally inducted into the Society on Friday morning. Dr. Kaelin is a Sidney Farber Professor of Medicine in the Department of Medicine at the Dana-Farber Cancer Institute at Harvard Medical School and an investigator of the Howard Hughes Medical Institute. Dr. Kaelin received his MD from Duke University and then became chief resident in internal medicine at Johns Hopkins. He was then a medical oncology clinical fellow at Dana-Farber and a postdoctoral fellow in the laboratory of Dr. David Livingston, where he began his studies of tumor suppressor proteins and their mutations. He then became an independent investigator at the Dana-Farber. Among his many awards, in 2019, Dr. Kaelin was awarded the Nobel Prize in Physiology or Medicine, along with Peter Ratcliffe and Greg Semenza, for his discoveries of how cells sense and adapt to oxygen. Let me just introduce him by saying that his work is just a beautiful illustration of the importance of basic research and how from studying a basic research problem can flow a whole range of medical applications, indeed medical applications which I'm sure have touched many of you and your families. So with that, let me just provide his title, The Case for Curiosity-Driven Research, New Treatments for Cancer and Anemia Emerging from Studies of an Obscure Hereditary Cancer Syndrome. Please join me in welcoming Bill Kaelin.
While we're waiting, back in the era of 35 millimeter slides and projectors, I used to say there were basically two things that could go wrong. Either the slide got jammed or the bulb burned out, and that was about it. Of course, in the world of PowerPoint, there's an infinite number of things that can go wrong, and we experienced about a dozen of them this morning, which is why we're changing computers, so we'll see how this works. Okay. Is the advancer working, or should I? Good? Okay, thank you. Okay, well, thank you very much. So when I was a young man, I thought I was going to be a clinical doctor. Here I am on the first day of internship at Johns Hopkins. My chairman was the great Victor McCusick, who not only was a member of the society, but also had won the Benjamin Franklin Medal. And he instilled in me an appreciation of the power of human genetics. Francis, I'm sure that would resonate with you, as well as the power of understanding the work that's been done before you, having some sense of medical history. I went, as you heard, to the Dana-Farber Cancer Institute, again, expecting to be a clinical oncologist, but I had the very good fortune of meeting the great David Livingstone, who sadly we lost 2 years ago. And it was David who inspired me to be a scientist and taught me how to be a scientist, so I really owe my scientific career to David Livingstone. Now, I understand this is a very sophisticated audience, but I suspect it's also a fairly diverse audience, and I always tell students no one's ever gotten mad at me because I made them feel smart. So we're going to have a 3-slide primer on molecular biology to make sure everyone's in the same boat. So if we recognize that a segment of DNA has the instructions for making a protein, we refer to that as a gene. You can think of that as the master blueprint. If the cell needs to make more of this protein, it makes copies of the DNA in the form of RNA. So you can think of these as the Xerox copies of the master blueprint, and then these get sent to the factory floor to make the protein using those instructions. And a protein, I like to think of as sort of a string of pearls, where each pearl is one of 20 potential building blocks, so-called amino acids. And it's the three-dimensional folding of that string that determines the function of the protein. And we represent these different building blocks with different letters, such as the one shown here. Now, the problem is sometimes the DNA sequence gets altered or mutated. This misinformation gets incorporated into the RNA, which in turn, at times, can lead to a disastrous change in the protein. So here, the building block represented by the C on the left has been changed to the building block represented by the F. Now that might seem like a subtle change, but this is literally a mutation which in certain families around the world predisposes them to various cancers. And then the last part of the primer is that genes can be, if you want, on or off, or scientists say expressed or not expressed. And so if the cell needs more copies of that protein, it will turn on the Xerox machine and send those blueprint copies down to the factory floor. And that's under the control of so-called transcription factors. Now, in David Livingstone's lab, I had worked on a so-called tumor suppressor gene, the retinoblastoma gene. This is a gene that contributes to cancer through its inactivation. And then, as you heard, I started my own laboratory in 1992. And shortly after starting my laboratory, this paper crossed my desk, which was the cloning or isolation of another so-called tumor suppressor gene, in this case, the von Hippel-Lindau tumor suppressor gene. And this is a gene that, when altered and passed down from generation to generation, can predispose individuals to various cancers. And I should point out that this work was led by investigators at the NIH, which sadly is currently under attack. Now, what do these families look like? So here is a number of these von Hippel-Lindau disease families that have been followed for years at the National Cancer Institute. Every filled circle, or square is an individual in that family that's developed one of several different types of cancer. And the most common cancers include kidney cancers, blood vessel tumors called hemangioblastomas of the eye, brain, and spinal cord, as well as so-called neural crest tumors, sometimes referred to as paragangliomas. Now, this disease affects about 1 in 35,000 people around the world. It was described by the German ophthalmologist Eugen von Hippel in 1904, and subsequently reported in greater detail by the Swedish neuropathologist Arvid Lindau. Now, just to look a little more closely, your DNA is organized into 23 pairs of chromosomes, but here I'm showing you the pair of chromosomes from mom and dad that harbors the VHL gene. So people with von Hippel-Lindau disease have inherited a defective version of the VHL gene from mom or dad. In this schematic, it's the maternal copy that's defective. But unfortunately, being born like this, they have a 90% chance that one or more susceptible cells in their body, such as in the kidney or in the eye, will spontaneously mutate or lose entirely the other normal copy. And that's the cell that's then at risk of going on to form a tumor. And as you would expect from the knowledge that inheriting a defective version of the VHL gene predisposes to, for example, kidney cancer, if you now look at garden variety non-hereditary kidney cancer, and the most common form of kidney cancer is so-called clear cell renal cell carcinoma, you again see that frequently both the maternal and the paternal copies of the VHL gene have been mutated or lost. But here, both mutational events, or hits, if you will, have occurred sometime after conception, in contrast to VHL disease, where the first mutation has been inherited from mom or dad. Now, I knew from my clinical training that the tumors seen in von Hippel-Lindau disease are notoriously rich in new blood vessels, and that's because they sometimes overproduce various growth factors, such as vascular endothelial growth factor, which acts as a fertilizer for blood vessels. And then another curiosity is they sometimes cause patients to make too many red blood cells, and that's because these tumors sometimes inappropriately produce a hormone called erythropoietin, which stimulates red cell production. Now, what VEGF and erythropoietin have in common is that they're normally induced when cells or tissues are not getting enough oxygen. And this makes sense. If you're not getting enough oxygen, you want to be able to improve oxygen delivery, you want to improve the oxygen carrying capacity of the blood by making more red blood cells, and you want to have more blood vessels supplying those red blood cells to the tissue in need. So this was the clue that by studying the VHL gene and its protein product, we might actually finally begin to understand how cells sense and respond to changes in oxygen, which seemed like an important problem, since oxygen plays such an important role in so many diseases, including cancer, but also heart attack, stroke, etc. Now, we knew from the work of many laboratories, including the work of my fellow laureates, Greg Semenza and Peter Ratcliffe, that many of these so-called hypoxia-inducible genes, and hypoxia is just a fancy word for low oxygen, such as VEGF and erythropoietin that I just mentioned, are under the control of a transcription factor, but you're now experts on transcription factors, that contains two subunits, the alpha subunit and the beta subunit. And so this is going to turn on the Xerox machine. But we also knew from their work that the alpha subunit is normally destroyed or degraded if oxygen is plentiful. It only becomes stable and active when oxygen levels are low, hence hypoxia-inducible factor. And we and others then went on to show, and I think this is hopefully the most complicated slide I'm going to show, that the VHL protein is actually part of a multi-protein complex that, in the presence of oxygen, chemically attaches a little flag made up of a mini protein called ubiquitin. And by adding this little flag, the HIF-alpha gets targeted for destruction by a cellular garbage disposal we refer to as the proteasome. Whereas when oxygen levels are low, or the VHL protein has been mutated, such as in kidney cancer, now HIF-alpha can accumulate, dimerize, or bind to its partner protein HIF-beta and activate genes such as VEGF. Now, this was very satisfying, but it still begged the question, how do cells know, if you will, whether oxygen is or is not available? How would the VHL protein know whether it should leave HIF-alpha alone or whether it should target it for destruction? And our lab and Sir Peter Ratcliffe's lab working in parallel showed that in the presence of oxygen, one of those building blocks within the HIF-alpha protein that we recognize by P for proline gets chemically modified. A little hydroxyl group gets added to the proline, and that serves as the signal that oxygen actually is available, and this then generates a binding site for the VHL protein. And what was very satisfying about this is even a sixth grader would appreciate that this O stands for oxygen. So this was an immediate link to oxygen availability. Now, of course, like most things in biology, it's a little bit more nuanced than that. We and others showed that the proteins that add that hydroxyl group, and by the way, if a protein accelerates a chemical reaction, we refer to it as an enzyme. So the enzymes that do this work, which have this name of EGLN, they split molecular oxygen, and they use one of the oxygen atoms to add the hydroxyl group. But importantly, they also have a fairly low oxygen affinity, so they're very sensitive to changes in oxygen availability in a physiologically relevant range. Now, these enzymes also require several other chemicals. They require iron, but I also want to point out they require this funny chemical over here called 2-oxyglutarate, which during the reaction gets modified and converted to succinate. Now, I point this out because when we first discovered this 20 years ago, it immediately became apparent that you could inhibit these enzymes with orally available drug-like molecules that would compete with 2-oxyglutarate and effectively shut these enzymes down. And once you shut the enzymes down, the body or the tissue thinks it's not getting enough oxygen and responds accordingly. So, for example, if you take this by pill form, your body thinks it's at the top of Mount Everest, and you start making your own red blood cells because you start turning on the genes dedicated to red blood cell production. So a number of pharmaceutical companies picked up on this and started to develop these orally available drugs that would basically act as hypoxia mimetics. You would now think you're not getting enough oxygen. And the initial indication was in anemia, where you're not making enough red blood cells. And there are millions of people around the planet who have anemia because of chronic kidney disease. And for that and other reasons, they don't make enough of their own red blood cells. Now, the first of these to be approved was a drug called daprodustat. I've been involved with a company called Fibrogen that was developing a drug called roxadustat, and I should declare I have a financial conflict of interest in roxadustat. Suffice it to say that this drug has been approved in over 44 countries around the world. Sadly for me, the US FDA decided they wanted yet one more phase 3 safety study, and the company figured out that by the time this trial ended, the drug was going to be off patent. So that's a whole other discussion. But importantly, for a variety of reasons, the first country to approve this drug was China, in part because of concerns about the reliability of their transfusion supply, et cetera, et cetera. And so as of last month, there are several 100,000 Chinese patients who take this drug, roxadustat, every day to treat their anemia as an alternative to transfusions and erythropoietin. Now, returning to the role of HIF in cancer, what I didn't tell you was HIF's actually a small family, and I show you the two most famous members of the family here, HIF1 alpha and HIF2 alpha. But work that we and others did showed that at least in kidney cancer, the culprit really is HIF2. And in fact, many kidney cancers get rid of HIF1 altogether. So what could we do about this therapeutically? Well, by the 90s, a number of drug companies were already making inhibitors of VEGF, and we argued that if VEGF inhibitors were going to work in any solid tumor, they should work in kidney cancer because of this direct and intimate connection between HIF and VEGF and VHL. And that turned out to be the case, so I think we're up to 8 or 9 VEGF inhibitors that are now approved for the treatment of kidney cancer. So that's the good news, but the bad news is not every patient with kidney cancer will respond to these drugs, and even those that do usually will progress within a year or two. So how can you do better? Well, I think you might argue based on first principles, rather than targeting any one single HIF2 target gene, maybe we should target HIF2 itself. But the conventional wisdom was that HIF2 wasn't druggable. It's an intracellular protein, and it was thought to lack the right kinds of nooks and crannies to drug with a small molecule. But fortunately, two wonderful scientists who were then at UT Southwestern, Rick Bruick and Kevin Gardner, ignored that dogma. They identified a potentially druggable pocket in the so-called PAS-B domain of HIF2 alpha, which is what I'm showing you here. They then, in their academic labs, identified a chemical that could bind to this pocket, and in so doing, cause the protein to refold slightly in such a way that it would no longer bind to DNA. Now, these were good for proof of principle, but they had many blemishes and warts that would have prevented them from being true drugs. But fortunately, they were out-licensed to a company called Peloton Therapeutics that was later acquired by Merck. And again, I declare another financial conflict of interest. But the medicinal chemists at Peloton made these chemicals much more drug-like. They improved their potency their specificity, and their bioavailability. Our lab and the lab of my former postdoc, Jim Bruguerolas, then showed that these first-generation HIF2 inhibitors were highly active in preclinical models of human kidney cancer. An improved version of this molecule called belzutifan then went into phase 3 testing and ultimately was approved for the treatment of advanced renal cell carcinoma. Now, a truism, and by the way, I cannot see a clock. I'm told there's a clock there, but this is dangerous. But I also speak New York fast, so I'm probably doing okay. So the drug was approved for sporadic, non-hereditary kidney cancer, but a truism in medical oncology is that most cancer drugs work better in a frontline setting than they do in very late lines of therapy. And so we were able to convince Peloton and then Merck to treat 61 patients with von Hippel-Lindau disease who had measurable kidney tumors that had never been treated before. These patients develop so many tumors over time that they're often put in surveillance programs where they'll have an MRI every 3 or 4 months. And only if a tumor changes in a worrisome way will they go to the operating room, whether that's a kidney tumor or an eye tumor or a spinal cord tumor. So these are so-called swimmers plots for obvious reasons. So here are the 61 patients. So each horizontal bar is a patient on the trial and how long they were on the trial with the HIF2 inhibitor at the time of this analysis. So for orientation, here's 1 year on therapy. All the patients with the black arrows were still doing well on therapy at 3 or 4 years. And every place you see a blue square or a green square, that patient's kidney tumor has actually shrunk or disappeared altogether. Now, if you prefer so-called waterfall plots, here's how the tumor shrinkage looks on a waterfall plot. So again, here are the 61 indicator tumors that allowed these patients to get on the trial. Every time you see the bar go below 0, the tumor is shrinking on the drug, and every time it goes above 0, it's getting larger. I'm a lumper, not a shrinker, not a splitter. So I would say all the tumors got somewhat smaller, but if you're a splitter and you're like RECIST criteria, the formal RECIST response rate was 60%. Now, to be on the trial, you had to have a measurable kidney tumor, but you could have any of the other tumors that VHL patients develop. So, for example, some of these patients had central nervous system hemangioblastomas, and you could see they responded very nicely. Some of those patients had those neuroendocrine tumors I talked about, and you can see those tumors responded as well. And perhaps most importantly, I mentioned these patients were in surveillance programs. So in gray are the 4 years of observation for these patients before they went on the HIF2 inhibitor in green. And every place you see a little geometrical symbol, that patient's going back to the operating room to have another tumor removed from their kidney, their eye, their spinal cord, the brain. And you can see that the surgeries, they don't go to 0, but they certainly decrease dramatically. And so based just on this phase 2 trial, in the 61 patients, the FDA did approve this treatment for von Hippel-Lindau disease almost 100 years after the initial clinical description. Now, it's very easy to look at clinical data and forget that there are actual human beings attached to all the data, at least it is sometimes for me. So I thought I would just show you one social media post. It was sort of fun. My kids taught me how to do this. I didn't have to wait for the New England Journal article. All I had to do was go to social media sites because I could see the patients were posting that they were responding. Because you can imagine if you have von Hippel-Lindau disease, you've had the sword of Damocles over your head. And you assume this disease was going to be your undoing. But here's a typical social media post. I never thought I'd see this day, and they're describing that their tumors were getting smaller or were stable, or in some cases had disappeared entirely. Now, in closing, I showed you this paper earlier, Eugen von Hippel's paper in 1904, but I also told you Victor McKusick taught me the power of understanding medical history, and it was also Victor McKusick who pointed out this was not, in hindsight, the first description of von Hippel-Lindau disease. The first description of von Hippel-Lindau disease actually occurred 10 years earlier from the great English geneticist Treacher Collins. So why was this paper largely overlooked? Why was it largely ignored? There was a simple reason. It was published in English. So this was the great descriptive era in medicine. And if you could only read one medical literature in the late 19th century, or the early 20th century, you read the German literature. So ironically, his undoing was he published in English. Now, what changed? Well, one thing that changed, of course, was World War II and the demise of Germany. But the other, I think, and I see this name comes up again later in the week, was the work of Vannevar Bush, who was also a member of this society. So Vannevar Bush, if you don't know, was arguably the key figure who, in the middle of the last century, convinced the federal government that the best thing they could do was to sort of pump prime by investing in basic and fundamental research in the academic sector, and then let the private sector decide when that knowledge had occasionally matured to the point where it was actually useful and could actually be applied. So this was absolutely brilliant. It served us well for decades. Now, I'd be the first to admit that Nobel Prizes don't necessarily perfectly capture a country's discovery power, but as a crude metric, you can see that during the descriptive era, when von Hippel and Lindau were publishing their papers, you could see the dominance of Germany, and then Vannevar Bush comes along, and something changes. And I think it's a combination of federal investment in basic and fundamental research, coupled, of course, with immigration, because we became this shining city on the hill, and especially with the brain drain and leaving war-torn parts of the country, people came here. Now, of course, we're all painfully aware that for some odd reason that escapes me, we're attempting to undo this. So this was my feeble attempt to add my voice to the many voices saying, what are we really doing here? Because again, we had a model which was the envy of the world, has served us well for decades, is responsible for our leadership in multiple innovation industries, including the biotech and pharma industry that I'm very close to, but certainly many other industries as well could map back to that investment in basic and fundamental research. So hopefully we can right the ship here, and I purposely don't want to leave you with a complete downer. So I'm always looking for something uplifting, something hopeful, so I'm going to just share one of many things that I think are hopeful. So this is a wonderful organization called the Environmental Voter Project. What they hypothesized and experimentally confirmed is that there are millions of Americans who never vote, who, had they voted, would have voted with the environment as their #1 issue. And these people, by and large, also believe in science and objective truth. Now, it also turns out how you vote is confidential. Whether you vote is not. So whether you vote is discoverable. So they can go into voter logs and look and see who votes, who doesn't vote. They've built incredibly accurate, robust analytical models to find these people. And by the way, if you think modern campaigning is about changing people's minds, that went out the days of the dodo. Modern campaigning is using analytics to find people who are already predisposed to vote the way you want them to vote, and then you use behavioral science methods to energize them and get them to vote. So this group actually does randomized clinical trials where they identify these non-voters. They then go in with these embarrassingly simple but effective behavioral science techniques, and they can actually map how many voters they have created in the last 10 years. So there are up to 2 million voters who never voted before they found them. They think they can get north of 5 million super voters, pro-environmentalists, pro-science, and at that point, you're more powerful than the NRA. Because the NRA is powerful simply because it's a galvanized block of voters that people can't ignore. So this is one path amongst many to try to get more people engaged in science, who believe in science, to actually vote. Because in the end, the solutions will have to be political. And my young people, friends and family tell me you have to have a QR code these days, so we even have a QR code. Thank you very much.
Thank you, Bill, for your work and for your comments. Questions? Francis. And state your name and where you're from.
Where I'm from. Well, let's see. I'm Francis Collins. I used to be at NIH. Bill, that was a great talk. Curious to know, with von Hippel-Lindau, obviously it's a 2-hit model, but for most cancers, it takes more than 2 hits to get all the way to a cancer. Is there any other sort of additional mutation, a collaborator that might also be a drug target?
Yeah, I'm glad you asked that, because you've anticipated the answer. So yes, absolutely. So like most common solid tumors, I would say VHL inactivation is the initial key step. Some people even call it the gatekeeper. But you absolutely need additional mutations in other genes before you'll go all the way to a kidney cancer. We know that not only from studying the von Hippel-Lindau disease patients, but also looking now with modern techniques at the non-hereditary kidney cancers. And so I like to use the analogy of tumblers on a lock. And for kidney cancer, I think the first critical tumbler to fall in place is VHL loss, but there are at least 3 more, and we can talk offline, but we know most of those genes as well. This turned out to be really critical in considering the safety or lack thereof of drugs that stabilized HIF, right? Because naysayers would say, well, you're just going to have von Hippel-Lindau disease in a pill because you're deregulating HIF. Now you're smiling because you know that's an oversimplification, but you won't be surprised to hear that that was said by some well-intentioned people. But part of the issue is, again, you do need multiple other tumblers to fall into place before you'll be a kidney cancer.
Jack, back there.
Jack Liu from East Lansing, Michigan. Thank you so much for this fantastic talk. I like everything you said, although I may not understand everything you said, but as an ecologist, I'm particularly like these slides. And because as an environment scientist, I think the interface between medical professional and scientist and the environment issues are hugely important. I'm wondering, how did you get, into these issues that I think are not just politically important, but also environmentally important? Thank you.
Yeah, so fortunately, I read the fellow who started this. His name's Nathaniel Stinnett, a very smart Yale grad who had worked in modern campaigning, knew how modern campaigns worked. And again, his epiphany was that if the environmental movement had the equivalent of the NRA, we'd be carbon neutral by now, because the politicians' behavior is so predictable and mappable, right? And he's the one who taught me things like, why the NRA does have so much power. And it's this galvanized block of voters. And he also taught me this simple fact that whether you vote is public. So he said, if you call your elected official complaining about something, the first thing they do is they have their assistant find out if you ever voted. If you've never voted, the conversation is over. It's over. Okay. If you do vote, they have very accurate data on what their constituents care about deeply. And for most of our lives, the environment has come in 8, 9, or 10, with 1, 2, 3 in some order being the economy, healthcare, and defense. So he realized he needed to move the environment up a bit. And again, I already told you the punchline. His hypothesis was absolutely confirmed that there are a lot of environmentalists who simply never vote. Now, the related question he gets asked, and I often get asked, is why aren't they voting? That's a whole other discussion, and I know we have some sociologists here. But there's a lot of overlap between these people. It's not, first of all, it's not a homogeneous group, but there's certainly a lot of overlap between these people and people who are historically suppressed from voting. So you may be solving 2 problems at once in terms of how do we get everyone engaged and voting.
Anyway, don't shoot me, but I'm a former U.S. Congressman, Patrick Murphy, but I'm on your team. So, the University of Pennsylvania. This is the last. I follow this pretty closely, but of the last 10 elections, the 9 out of 10 were change elections, where either the White House, the House, or Senate changed. I love the fact that you're not saving this conversation for Play Company, that you're challenging all of us. Appreciate it. I would say to your thesis on this is that as long as you look like you're being part of the solution, saying, hey, listen, we need to change this, we need to do better, but look at the great success. I mean, I even wrote it down, that we're killing the golden goose. So what do you think we need to do better as an organization at APS that cares about this, beside joining the environmental voter org, beside showing up today? Because it's easy to talk to the choir, but not the congregation.
Yes.
What would you say we should do publicly? I love the fact that you're on social media, but what other advice would you give us?
Yeah. Well, thank you. First of all, I actually like that you used the word team, because when I was a young boy, I didn't think this was a partisan issue. And when I give a longer version of this talk to young students thinking about a career in scientists, I often my first slide is usually Sputnik, since I was born in 1957. But in the 60s, 70s, and even largely in the 80s, it just was unfathomable that this ever would become a political issue. I thought we were all on the same team. And so how we get people to understand that some things should not be about partisanship, that we all get affected by these diseases, right? I wrote an editorial when my wife died of glioblastoma. She died about the same time as Senator McCain died of his glioblastoma. And I point out this is not a team thing. I mean, it's a team in that we're all in the same team. It's not Republican versus Democrat. But what happened during the 60s? Precisely because of Sputnik and the Cold War, there was bipartisan realization that this was almost existential that we win when it came to science, technology, and engineering. So, when Obama said we need another Sputnik moment, maybe we need another Sputnik moment, but we need something like that. I mean, people have to understand that whether they're scientists or not, we're all in this together, and they all benefit from us being a center of innovation. And I can go on and on. So I'm preaching to the choir, but I think it's partly education, outreach, as you said, crossing over when we can to our colleagues on the other side.
But we did have the COVID epidemic.
Yes.
And in fact, that separated us rather than bringing us together.
Yeah. Well, we were talking about this a little bit at lunch. I think sadly, and maybe it's in the unintended consequence book that we learned about this morning, I think people who are smarter about this than I am have pointed out that we're paying the price for people who, when it was expedient to do so, sowed distrust in the scientific method, whether it was people who made money from a carbon-based economy pushing back against climate change, whether it was the tobacco industry pushing back against the cancer risk, whether it was people who wanted to add lead to everything, including your gasoline and your paint. The playbook was always the same. Question whether anything is objectifiably true, whether there really is such a thing as expertise. It's just a theory, it's your opinion. But we're paying that in spades, and I think the same playbook unfortunately played out a bit in COVID, although there was a bit of a stretch. Why? I don't completely understand, but I think that's where we got it. I'm sure part of it was the wishful thinking that we could ignore this and go back to work, because of course it was costly on multiple levels, right, to shut down.
Yep.
And so I'm sure there was some of the same economic interests, again, might have been behind some of it.
All right. Well, with that, let's thank Bill again. We're now going to take a 20-minute break and then return for another part of the great session. Thank you.
A little while ago. She should be coming back in a short.
If everybody can please return to their seats. We need another ringing of the bell. Can everyone please return to their seats? Thank you. Can everyone please return to their seats so we can continue with the session? We need a bell ringing. Thank you. Thank you. It's time for everyone to sit down. Please return to your seats. And of course, please turn off your cell phones. Okay. All right, thank you. Thanks very much. So our next two talks will be interesting to be thinking about in the context of things going on in Washington currently. So our next speaker is Michèle Lamont. She's also recently elected member of the APS and just inducted into the APS earlier this afternoon. Dr. Lamont is the Robert Goldman Professor of European Studies and a Professor of Sociology and African American Studies at Harvard. She's a comparative sociologist and contributor to the study of culture, inequality, racism, and anti-racism. She obtained her bachelor and master degrees at the University of Ottawa and PhD in sociology from the Sorbonne. After a postdoctoral fellowship at Stanford and faculty positions at UT Austin and Princeton, she assumed her faculty position at Harvard. Her research addresses how we evaluate social worth across societies and the role of cultural processes in fostering inequality. Among many awards, in 2024, Dr. Lamont was named the Coley Prize Laureate by the Coley Foundation for Sociology in recognition of her studies of social boundaries and social worth, highlighting the foundations of conflict and integration in contemporary society. She's received honorary degrees from 5 countries, and she's been elected to the British Academy and the Royal Society of Canada, among others. Her talk today is titled, Seeing Others, How Recognition Works and How It Can Help Heal a Divided World. Please join me in welcoming Michèle Lamont.
Thank you so much. Thank you for this nice, very kind introduction. So it's a great honor to be here today, and I'm really humbled, especially when I read the bios of the other inductees today. So I spent a good part of my life trying to understand how boundaries between groups can be made more permeable. And shortly after Trump was elected in 2016, one of his very first executive act after his election was to pass a travel ban toward Muslim countries and also other decisions having to do with reducing the rights of trans people. And as a cultural sociologist, someone who studies the interaction between culture and inequality, I thought I know exactly what is happening there. It's making these boundaries more rigid. And of course, this is very upsetting to many people, but the average citizen doesn't necessarily have the language to understand these mechanisms by which boundaries are made more rigid. So I thought I should write a book that would help the general public understand how boundaries work, how these definitions of work can be transformed. Now we hear a lot of calls for greater empathy, but in fact these are cultural processes, and I wanted the general public to understand this. So I decided to write this book, which came out in September of 2023, titled Seeing Others. And to explain to you what this book is, I show you two covers. The American version, Simon & Schuster, you have the circle of people who feel that they belong, which I call cultural membership. They're worthy, they're listened to, they are defended by others. And you have groups of different size and different colors that are further apart from the center, or wanting to be in, if you want. So this design really shows very clearly what this book is about, whereas the British subtitle, which is different from the American subtitle, really captures what the book is about, how to redefine worth in a divided world. So the context in which I wrote this book, I started writing it, well, actually doing the research in 2019, is one where you had an explosion of social movements, which all had to do with recognition claims. So these are groups that feel they have been treated unfairly, their dignity has not been respected, so they take the street to protest. And this is happening at both ends of the political spectrum, and it's happening in Europe as well as in North America. So you have Black Lives Matter and Me Too. You also have MAGA, which is often associated with working class men who have been downwardly mobile and who are embracing nationalism, a high status characteristic, their American nationalism, to lift up their worth. You have the farmers in Europe. So for me, this was also a moment where I was slightly depressed, like many other people, and I decided to look at the literature, the social science literature on hope. There's a very interesting literature that's been growing rapidly on how people imagine the future, which has everything to do with hope. If you feel like you're in a dead end, if you cannot think about alternative, it's easy to be depressed. And what that literature says is that this can very much be influenced by the narratives to which you are exposed. The field in which I work, cultural sociology, since the mid-'80s, has really focused on the analysis of the scripts that are available to us. These tools that are made available to us in our environment on which we can draw to make sense of what's happening to us. The meaning-making process is very much influenced by the cultural structures, we call them cultural repertoires, in which we live. So an alternative to simply telling people be empathic would be to try to think about how the cultural structures in which we live can be shaped differently in such a way that they're becoming more inclusive and less focused on what someone like Jonathan Haidt calls tribalism, which is the notion that we are wired to like people who are like us and dislike people who are not like us. And that's tied to an argument about human nature. I don't know anything about human nature. I'm interested in how can we structure our environment in such a way that people are more likely to define group boundaries in a much more fluid way. So that's my intellectual agenda in this book. So to study the transformation of repertoires, I decided to conduct in-depth interviews. That's the methodology that I typically use with people whose job it is to create narratives. So it was the pandemic, great time to conduct interviews on Zoom. No one was working. So we decided with my research assistant to look at people whose job it is to create narrative and people who are extremely influential in doing so. We were able to get hold of a lot of people who are working in Hollywood. One of these people is Joy Soloway, who created the show Transparent. Some of you may be familiar with it. It was on Netflix, very popular. And it tells the story of a woman who are advanced middle age, is a trans woman. And she has, after having transformed her identity, she has to negotiate this new identity with her adult children. So when I interviewed Joy Solway, they explained to me that the reason they were doing this show is to make trans people less abhorrent, more understandable, and also to show trans people in more multidimensional way, less stereotypical way. And these are words that we heard time and time again with all the interviews that we conducted, which have to do with recognition, with destigmatization. So the T-shirt, I see you, this is an expression that became extremely popular with Gen Zs. And so the book is in part with people who are doing creating narrative that become very influential and diffuse broadly in society, but we also look at the problem from the bottom up by interviewing Gen Zs who are receiving these messages, but also trying to think of their life differently. What are the challenges that they're encountering, how they would like to live their life differently. The other T-shirt is, of course, something that many of you recognize. It became a very popular poster on many lawns in the US during the pandemic as well, and it embraces what I called ordinary universalism. The slogan, "Love is love," became very important in the fight for same-sex marriage, and of course, "No human is illegal" became very salient in the context of the defense of undocumented immigrants. So the broader agenda is also to think about social resilience as something that is built through the institutions that we create and the cultural repertoires that structures our environment. So an example of how social resilience results from institutions would be same-sex marriage laws that were not passed to provide messages about recognition, but they had a direct effect on a decline in attempted suicide among LGBTQ high school students. So it's a case where the production of meaning and their diffusion, broad diffusion, ends up providing messages to groups that they are worthy, and it has a direct impact on their well-being. So the key concepts here are the concept of recognition. It's been associated with Hegel, but more in the last decades, there's been an important debate between two philosophers, Axel Honneth and Nancy Fraser, which had to do with the politics of distribution recognition. Should politics focus on the distribution of resources or the affirmation of people's identity and dignity? So Honneth provides a definition of recognition as a social act by which the positive social worth of individuals or group are affirmed. So what's important here is that it's intersubjective. It's something that other human beings can give you, and it's a social act. It's something that we accomplish as we interact with each other. So it's not inborn, it's really a cultural process. It's often talked about in relation to stigmatization, which is just the opposite, defining people as unworthy. And those two cultural processes operate all the time as we lead our life. And they result in definitions of cultural membership, where different groups are defined as more or less worthy, and with this comes a shared understanding about our obligations toward other people. So a group that is deeply stigmatized, we will not think that we owe them anything. So for instance, the notion that we should not support drug addicts or, homeless people or immigrants or undocumented immigrants would be associated with this. And these, So when it comes to the actual research, we did 185 interviews with these change agents, as well as 80 interviews with college students, both before and during the pandemic. And we tried to capture what kind of messages that they were trying to produce and why. And the context is really, decades of growing inequality where the American dream is in deep crisis. We know that millennials, after 2008, very much felt like the American dream was not for them. We've had a major mental health crisis among Gen Zs. All of us who teach undergrads know that this is quite acute. So they really like grasping for an alternative. And what we found through our interviews is what's the content of the alternatives, which is what I'm going to talk to you about. So this required thinking about who are the agents who are really transforming the narratives in which we live. What are the new narratives being produced? So we identified a number of organizational fields that play an important role. So that's only some of them. The universities should be there as well. And we reached out very broadly through various networks to identify people who would be interested in being interviewed. And we asked them questions precisely of the kind that I asked Soloway about Transparent. What are you doing? What are the messages? So 75 interviews with stand-up comic, for instance, where we develop a typology of how they talked about the messages they want to diffuse. Some of them said, our job is not to force people to eat their vegetables. We want to get them to laugh, but at the same time, we plant little jokes about racist or about sexist people or about, so there's a lot of implicit meanings that are being diffused. So here you have some of the people we were able to interview. For instance, Michael Schur, who created Park and Recreation and The Good Life, a lot of people who were really, really influential. Shining Donghae, who was a director of Netflix at the global level and decided to diffuse Black Panther in Africa and India. African-American woman, when I interviewed her, she said, I really wanted to put Black Panther to make it available in Africa so that the young African kids could see this story, a very epic story of a successful, African society. So there was a lot of consistency of what they were talking about. So the book, sorry, like the private speaker, the president speaker, I will say I'm talking New York fast here, so. It's a very short period of time to provide a lot of content, so. The book as a penultimate chapter, which is really to tell the general reader what they can do to contribute to making group boundaries more permeable. So one of them is to try to think about how do we move from a shared definition of who's worthy that tends to be very unidimensional, based basically on a very dominant conception of success, that we should all be college-educated professionals, toward a much more multidimensional understanding, pluralistic understanding that also allows people who do not have the college degree that has become, the basis for a caste system in the US, to also think of themselves as successful. The book also talks about how stigma has been reduced in the past. I provide the case of people with HIV/AIDS and compare it with people with obesity. The first group has become very destigmatized since the beginning of the HIV/AIDS crisis in the 1980s, whereas this has not happened with obesity. So we ask ourselves, if we look at this template, we understand how it has happened in the past, how can it happen more? So again, it's really an argument that the transformation of these boundaries is something on which we can have an impact. And the third aspect is ordinary universalism that is very much embraced by Gen Zs, the Gen Zs we interviews. And that's often called woke culture, but what characterizes it is that these young people are very much committed to leading their best life now because they don't believe in the American dream, and therefore it means that they want to lead their life today in a way that is very authentic with who they think they are. And they also embrace inclusion. So that's why the non-binary, for pronouns and the non-binary restroom become so important to them because they want to affirm people's rights to be who they are. So if I had more time, I would give you a lot of evidence about these three processes, but I'll see what I can achieve in the time that is left. So the common dominant understanding of who's a worthy self, as I said, are people with a college degree who can consume. People in this room would be example of this, and it comes with a focus on self-reliance, competitiveness, meritocracy, but we know that meritocracy is very much of a myth to the extent my children have two parents who have a PhD. They're born on an escalator. Of course, they can't demonstrate hard work and persistence, but nevertheless, they're born with such advantages. And we can see, looking at surveys, that belief in meritocracy has increased exponentially across all advanced industrial societies since the 1930s. So it's a myth, but at the same time, one of the big problems with meritocracy is that it really leads to a society that is increasingly bifurcated because the winners, people like us, upper middle class people, are constantly depicted in the media as worthy. Someone did the content analysis of entertainment television for the last 50 years, and he shows very clearly that there's very few working class people who are presented in entertainment television as worthy people. They're also always, ridiculous. Bart Simpson, the father, is a total idiot. So in this context, it's very difficult for working class people to ever think that anyone respects them. And that has operated hand in hand with growing inequality, desindustrialization, to feed the death by despair that Anne Case and Angus Deaton have written about, really aggravated conditions in the context that the working class has not only lost a lot of speed in terms of their purchasing power, but also they're becoming much more deeply stigmatized. And this, of course, is feeding the mega movement. So against this background, let's see how we can attack this situation that is so negative. So I'll just give you one example. There's one of the artists I interviewed, Hank Willis Thomas, did this video where he asked 130 black men across the country, what does it mean for you to be a black man? And he discovers they have absolutely nothing in common but the fact that they experience microaggressions all the time. And this video, which you can see on YouTube, was then used by a philanthropy and was projected in over 2,000 community centers and high schools across the country to create conversations. And the goal of this conversation was precisely to move toward destigmatizing black men. So that's one example among many, but it's a very important one because it illustrates something that the book focuses on, which is what I call recognition chains, these kinds of collaboration that connect the cultural producers who create new narrative and the philanthropies that play a crucial role in scaling up the messages and making them widely diffused. Ford Foundation has played a very crucial role in this because as soon as Darren Walker became the president of the foundation in the early 2020 and 2013, he said, we're going to fight inequality through 4 pillars. One of those pillars is what he called, it doesn't matter, but it's a focus on narratives. And for instance, they sponsored the film Roma, which was shown on Netflix, which won an Oscar. And what the Roma is about 2 domestic workers in Mexico City. One of them is thinking of having an abortion. And what the film does is really humanize and really destigmatize, make them visible. It's not only about destigmatizing, it's also about making them visible. So that's an example of destigmatization. So all this to say, I'm wrapping this up now, it's about really diffusing a much more pluralistic understanding of who's worthy. And now I'm not going to have time to talk much about the other 2 processes, but I'm just going to say what we know about the destigmatization of people with HIV/AIDS is that knowledge producers, all of us, journalists, social scientists, legal researchers, medical researchers, we all have played a crucial role in diffusing the notion that anyone can have AIDS. At the beginning, it was viewed as the disease of gay men who spend too much time in public restroom having sex with all kinds of random people. Now we know that this is not the case at all. And whereas people who are obese are still viewed as basically lazy people who are not able to take care of themselves. So we can really trace the career of such stigma to understand how groups that have historically been stigmatized are not anymore. How does this happen? And finally, the Gen Zs. There's a chapter of the book that is on Gen Zs, where we look at how do they think a good life looks like, and we show that they're braiding different repertoires that had not been as present. I'm a boomer. You know, these young people are connecting. They're young Americans. They think you have to be agentic, take your life in your hand, be an entrepreneur, put together a life. But they talk a lot about the work-life balance, which was absolutely not central to the life of boomers. Those of us in the room will know. And they are also embracing this ordinary universalism, this woke culture that I talked about. So the result, and the book is very much about hope, because what it tells us is that, here I'm using the metaphor of 1,000 points of light, all these profusion of narratives is happening. It has happened during the period when I did the research. That was a fact. You might think, what is she talking about? Now we have Trump in power. Why should we be helpful? Well, these 1,000 points of lights are still happening. People are still producing all these narratives. Of course, there's a tug of war in the public sphere about which ideas are going to get most traction. And we had pre-pandemic, before Trump's election, posters such as this one that were very much all over the lawns again, across the country, defending undocumented immigrants. Well, we have really a lot of resistance that is bubbling up right now, as we saw over the last 2 weekends with the large demonstration. Trump has done all kinds of things to attack the very agents of change, the kinds of people that I've studied, but at the same time, and I just published earlier this week, this article in Salon that describes the mobilization across the red state to fight back against Trump's attack toward the NEH and other organizations that are precisely focused often on creating a narrative. So the takeaway would really be that, you know, this process of definition of worth is happening all the time, and every social movement you will always have reaction, counter-reaction to any social movement. It will happen again. We simply, you know, I think have to stand. So you will ask also, well, what do we know about these college-educated Gen Zs we interviewed are very progressive. It's a generation that very much defines itself through its progressivism. Between May and December, I've been involved in a research project that focuses precisely on the Gen Zs who are not in college, the non-college educated youth. We're comparing workers in Manchester, New Hampshire, and Manchester, UK. And we're focused precisely on how they gain recognition through politics, with a lot of them staying away from it because they think it's an absolutely corrupted world. They think it's all drama, it's all anger, there's nothing to be gained from it. But some gain recognition through the MAGA movement because Trump claims to be really serving them. But I'm defining all of this to you to see research. We're now turning toward the other end of the political spectrum to understand the definition of worth as it operates toward the more conservative end of the political spectrum. So there's so much more that I could say. We have a few minutes for questions, but I thank you for your attention. And we can discuss a little bit. Thank you.
Thanks so much. I'm sure Michèle will be happy to answer questions. Balcony, do we have a microphone up there? Okay. It's coming around. Thank you, Michèle.
There's like 6 hands up there.
Hi, thank you. Michèle, my name is Nina Jablonski. I'm from State College, Pennsylvania. Your work speaks to the increasing the porosity of established groups, regardless of how those groups were formed. Sometimes, in fact, in many cases, groups were not formed with any kind of consent or with any real evidence, such as racial groups in most parts of the world. I study the evolution of skin and skin color and color-based race. And around the world, dark-skinned people face incredible stigmatization and peripheralization simply because of being labeled as inferior because of their dark skin. How can your research on group increasing group porosity and reducing stigmatization actually help us reverse or somehow change the groupness of those inadequately and inaccurately defined groups?
That's a great question. So this concept of groupness depends on a number of things, including the cultural repertoires that are dominant in the context in which they live. And this same process of stigmatization and recognition I talked about. Those of you who don't know, I'm French-Canadian. I grew up just next to Ottawa when I was growing up. The main divide in Canada was it was linguistic. It was not racial at all. It was, in the past, it was religion in the 50s. And before it was religion, then it became linguistic. But now in Canada, people are very aware of First Nation who were absolutely invisible when I was growing up. They were, important communities living 2 hours north of where I grew up. No one knew about them. And now they are widely discussed in the Canadian context. We've had the Truth and Reconciliation Commission. Same thing with Black Canadians. They were absolutely invisible. Now we have growing awareness. So I think the phenotype is always, a number of markers can become very salient, and what becomes salient depend on history, certainly, but also there was we could talk at length about, the constitutional crisis in Canada, the transformation of immigration. So I think there's nothing inherent. I learned, for instance, that albinos in some African contexts, this is really the group the most stigmatized, and they would not be stigmatized in the North American context. They are viewed as magical. So we really, this is a social science question that we can actually study, and it's not based on absolute understanding of color, skin color.
Other questions? In the back there, maybe? Below?
Hi, Michelle. Thanks for a very interesting talk. I think I have to get your book.
Also up.
Yeah, there's a young man there, too. Yes.
Yeah. So you had an alignment of conservatives with not going to college and progressives with going to college.
Yeah.
I do know a few conservatives who have gone to college, and I wonder what effect they might have on your study.
Yeah.
Your conclusion.
Well, my study is a study of cultural production. It's not a study of cultural reception. The part of the book that is concerned with Gen Zs, we studied them as cultural producers as much as we studied them as, to capture their... We have questions about what they consume, but that's really not the center of it. So the book uses survey data throughout. I'm not claiming that, but we know, looking at the profile of the voters of the Democratic or the Republican Party right now, that the non-college educated have moved massively toward the Republican Party. So, this is a very well-established finding. So that's not, your question is excellent, but the purpose of I'm not a survey researcher, I'm not even an electoral expert, but right now I'm very interested in how the non-college educated youth, which is a group that is not studied at all, understand politics. And what we discover in our interview is that they really tie moral boundaries with class boundaries. So when you tell them what are the words you associate with politics, they say corruption. So it's all moral boundaries directed at people above. So it has to do very much with recognition, but that's a framework that political scientists don't use at all. They are described as low information voters, people, I'm not being overly critical of political science here, but the model of the informed voter is so much at the center of much of the work in electoral politics. So I think there's really room for much more collaboration between our discipline, because people like me, I study moral boundaries. It's one of my expertise, and I think moral boundaries, everything that Trump is doing is very much about, moral condemning of the Latin, Central American immigrants. Morality is all over the place, and that's how he speaks to his supporters all the time. And the idea that, what he sells to his supporters is, I'll give you recognition by making sure that all the resources come to people like us, because there's a scarcity of resource. So therefore, we eliminate everyone else who is needy and focus on our needs. Therefore, Ukraine, Gaza, trans, undocumented immigrants. So there's a lot of logics to it, which I call recognition through exclusion, whereas those who support the Democrats do recognition through inclusion, and often they tie this to their identity as people of color and immigrants among the people we interviewed in New Hampshire. I'm sorry for the long response.
No, quite all right. Thank you. I think, yeah, one final question, if someone has a microphone in the back, and please identify yourself.
Susan, can you hear me? Hello, Susan Fisk, retired from Princeton University, now in Jamaica, Vermont. So I'm very interested in the way you understand recognition and its relationship to porosity. So it seems like there's a mix of processes going on here. Part of it is recognizing your definition of yourself as a group member and a category, but not making that overly rigid. And is it about respect, or is it about trust and morality? So maybe you could elaborate a little bit on exactly what it is.
Sure. So thank you so much for this question, Susan. That's where my field and yours overlap. And your work has been extremely important in this line of research. In the UK, especially, you have social identity theory that has been extremely influential and is also influential in the US to a large degree. In sociology, the work of Richard Jenkins, which I did not reference, but which I use in my talk, this dynamic between social how other groups define your group, which is called group categorization, and self-identification. What is the meaning of, what meaning do I associate with myself? And those two elements are constantly at play. So these questions are studied, approached slightly differently across the discipline, but our disciplines certainly converge there. We're much less focused on the micro processes, people like me, because the interaction with the process, the way we understand groupness, if you're interested in my view of this, it would be a book that I co-authored titled Getting Respect, Response to Stigmatization and Discrimination in the US, Brazil, and Israel, where we have groups that are stigmatized in the three countries: blacks in the US, blacks in Brazil, blacks in Israel, Ethiopian Jews, plus two other groups in Israel that are stigmatized: Mizrahim, Oriental Jews, and Palestinian citizens of Israel. And we interviewed them on how they experience discrimination and respond to it, and we show that the patterns are very much connected to how the groupness of each of these groups is expressed in a context where the dominant ideologies, the American dream, racial democracy in Brazil, frame their integration in radically different terms. So those are the things that sociologists really focus on that would not be part as much of the way that the question is framed by psychologists. But there's a lot of overlap there.
Thank you. And again,
Welcome.
thanks so
Thank you.
much.
Thanks.
So our final speaker of the day is Jeffrey Rosen. He's our neighbor, a neighbor to the APS as the president and CEO of the National Constitution Center here in Philadelphia, a professor of law at the George Washington University Law School, and a contributing editor to The Atlantic. Jeff Rosen has led the Constitution Center since 2013, fostering a bipartisan environment for debate and education in the U.S. Constitution. Rosen is a graduate of Harvard College and Oxford University, where he was a Marshall Scholar, as well as the Yale Law School. Under Rosen's tenure, the Constitution Center completed an interactive Constitution project which allows online visitors to examine contrasting interpretations of each clause of the Constitution and to compare the Bill of Rights to other constitutional systems around the world. Jeff Rosen also hosts the weekly podcast, We the People, for the Constitution Center, bringing scholars together to discuss timely constitutional issues and constitutional debates. His most recent book is The Pursuit of Happiness: How Classical Writers on Virtue Inspired the Lives of the Founders and Defined America. The book was published in 2024, and it's the topic of today's talk, and there will be a book signing after the talk outside. Please join me in welcoming Jeffrey Rosen.
So much.
Hello, friends. It is a great honor to be here at the American Philosophical Society. The Philosophical Society, of course, was founded by Ben Franklin in 1743. It was led by Thomas Jefferson from 1797 to 1814. Jefferson considered his election to the Philosophical Society the most flattering incident of my life, and he kept up his presidency while running the United States on the side in the early 1800s. I'm excited to be here because I want to talk with you about an unusual COVID-era reading project that led me to explore what Benjamin Franklin and Thomas Jefferson meant when they put that famous phrase in the Declaration of Independence about the pursuit of happiness. And what I learned changed my life. It changed what I think about how to be a good person, about how to be a good citizen, and also how to be a champion for civic education in America. Because I learned that for Franklin and for Jefferson and the other founders, the pursuit of happiness meant not feeling good, but being good. Not the pursuit of immediate pleasure, but the pursuit of long-term virtue. And by virtue, they meant self-improvement, character improvement, lifelong learning, using your powers of reason to moderate or modulate your unreasonable passions and emotions so you could achieve the calm tranquility that allows you to achieve self-mastery and to serve others. In other words, Franklin and Jefferson understood by the pursuit of happiness exactly the same sort of lifelong learning for which they founded the American Philosophical Society, which was designed as a society to inspire communication in the arts and sciences and to promote the cultivation of virtue. So let me tell you about this exciting intellectual journey that I've experienced, and then think with you about what the American Philosophical Society can do with the National Constitution Center and others to inspire civic learning in these challenging times. So here's how the reading project evolved. I noticed during COVID that both Franklin and Jefferson had chosen as their illustration of the central meaning of the pursuit of happiness a book by Cicero that I'd never heard of called the Tusculan Disputations. I discovered the Franklin connection when rereading the part of his autobiography where he talked about his efforts in his 20s to achieve moral perfection. And as you all know from this great project, he made a list of virtues and resolved to conduct self-accounting to see how he lived up to them every day. They included temperance, prudence, courage, resolution, cleanliness. He saved the one he found hardest for last, humility. Ba-dum-bum. And he made a graph with the virtues on the x-axis and the days of the week on the y-axis, and he resolved each night to put an X mark next to the virtue where he fell short. He tried this for a while. He found it's incredibly depressing because there are so many X marks. And he gave it up after a few weeks, but he felt that he was better for having tried. I knew about the project. I'd tried it with a friend in DC a few years ago at the suggestion of a local rabbi who recommended the Hebrew version. It was translated into Hebrew by a Hasidic rabbi who admired Franklin in 1808, and it's practiced today as part of the Musar, or character improvement system of Jews. But what I didn't know is that Franklin had chosen as his motto Cicero's Tusculan Disputations in a passage that said, "Without virtue, happiness cannot be." A few weeks later, I was at the Boar's Head Inn in Charlottesville on the beautiful UVA campus. On the wall of a hotel were a list of 12 virtues or canons of conduct that Jefferson had drafted for his daughters. They look a lot like Franklin's. They include silence, when angry, count to 10, when very angry, 100. And resolution, resolve to do what you ought and do what you resolve. Jefferson oddly left one of Franklin's 13 virtues off his list, and that was chastity. But what struck me was that when Jefferson was asked to illustrate his list, he would also hand out a passage from Cicero's Tusculan Disputations, a longer passage. He'd give it to anyone who asked when he was old about the central meaning of happiness. And he copied it out in his commonplace book when his dad died when he was 14 years old. And the passage from Cicero's Manual on Grief, isn't it remarkable that the central text for happiness in the founding era was a manual about overcoming grief? And Cicero had written it when his own daughter Tullia died, and Jefferson is copying it when his dad dies. The passage says, "He whose soul is tranquilized by restraint and consistency and is at peace with himself, who is neither unduly exuberant or unduly despondent. He is the calm and wise man of whom we are in quest. He is the happy man. So I thought, I've got to read this Cicero because it's so important to Franklin and Jefferson in defining happiness as tranquility of soul and virtue. But what else to read? Then I found this extraordinary reading list that Jefferson would hand out to anyone who asked when he was old how to be educated. And he'd give this to kids who were going to law school or to his friends who were building a private library. It's remarkable both for the scope of the books and for its schedule. The scope includes natural philosophy and astronomy and science and novels. Jefferson loves Richardson and Lawrence Sterne. What caught my eye was the section on moral philosophy. At the top of this section, sometimes he called it natural religion or ethics, Cicero's Tusculan Disputations, then Cicero's On Duties, then Epictetus, Aurelius's Meditations, Seneca's Letters, and Enlightenment moral philosophers, Locke's essay concerning human understanding, Hutcheson, Bolingbroke, Kame, and Hume. What struck me about the list is that I hadn't read any of the books on it. And I've had an extraordinary liberal arts education, and I give thanks every day to the magnificent teachers of literature. I was an English major in college, and history and political philosophy and law who inspired me and continue to inspire me today. But I hadn't read these books of moral philosophy because they'd fallen out of the curriculum when I was in college in the 80s. And I remember yearning for this kind of guidance. I was studying the Puritans with Sack Van Berkovich, the great Puritan scholar who described how the American Jeremiad has defined so much of our rhetorical history, and how just as the ancient Israelites were denounced for forsaking God's covenant, so the Puritan preachers denounced the Puritans for forsaking the word of the Lord, and so the early revolutionaries denounced the Boston congregation for going the way of Greece and Rome by surrendering their liberty for cheap luxury, but there's always the promise of hope and redemption. And I was studying Puritan theology at the same time, and I found myself unsatisfied by the notion that salvation could only be obtained by, not even by faith or good works, but by God's randomly given grace, which couldn't be affected by any amount of human will. This wasn't doing it for me, first because I'm Jewish, and also because it seemed unsatisfying for a life dedicated to seek virtue by reason rather than by faith or grace. And what I didn't realize, because these great books had fallen out of the curriculum, is this is exactly, of course, the question the classical moral philosophers set out to answer. The other thing that inspired me about Jefferson's list is the schedule. He said, you got to get up every morning before dawn, spend 2 hours reading moral philosophy, watch the sunrise, then political philosophy and history in the morning while the mind is still fresh, then lunch, then you save the science and math for the early afternoon until dinner, then you're allowed some Shakespeare and poetry for mild amusement and rational joy, and then to bed 12 hours a day, 7 days a week, no breaks. So I don't know what got into me. It was COVID. I thought I've got more time. I've got to read these books. It was really just to fill a gap in my education. I tried the schedule, not the whole thing, but the morning. I got up every morning 2 hours before the sunrise. I read the moral philosophy. I watched the sunrise every day, which remains the most spectacularly beautiful gift that you can give yourself any day. I found myself writing these sonnets to sum up the wisdom of the morning reading, which is incredibly weird, but it turns out all sorts of people in the founding era wrote sonnets to sum up the classical wisdom, including the great Phyllis Wheatley, the first published black poet whose genius was acclaimed by Washington, and John Quincy Adams, who would read Tusculan's Disputations in the original, his favorite book, in the White House and the Bible, write astonishing sonnets, including of abolitionism, and then walk along the Potomac. So that's what I did for a year, with no other goal than just to fill a gap in my education. But the reading changed my life, and I did indeed learn that for the founders, happiness meant self-mastery, virtue, self-improvement, character improvement. Today we might call it emotional intelligence, but it's essentially being a lifelong learner. But in particular, reading the moral philosophers helped me understand the moral psychology at the core of the project. Much of it went back to Pythagoras. I didn't know that in addition to inventing the triangle and the harmonic system, Pythagoras was a guru who lived on the island of Croton, who counseled rigorous vegetarianism and moderation in all things, and who distinguished the soul into three faculties or powers: reason in the head, passion or emotion in the heart, and desire in the stomach, and said that our goal is to use our powers of reason to moderate or modulate our unreasonable passions and emotions so we can achieve that calm tranquility of soul that later philosophers said defined virtue. I understood in a new light the chart at the end of the Nicomachean Ethics. Remember, Aristotle famously defines happiness as an activity of the soul in conformity with excellence or virtue. Eudaimonia, good demon, eudaimonian ethics. But I couldn't get it into my head what you're supposed to do until you see the chart at the end, and for every passion or emotion, there's an excess and a deficiency. So the goal is not exuberance or despondency, but moderation. And I now see that it's essentially a psychological product to avoid the perturbations of the mind, not to be distracted by perseverating about the past and the future, and not to allow yourself to indulge in unproductive emotions like anger, jealousy, ambition, and fear, so you can achieve productive emotions like the classical virtues, prudence, temperance, courage, and justice, and attain the tranquility that defines virtue and happiness. It also came home to me with two modern examples. It's a version of the marshmallow test at Stanford. The kids who were asked, if they want one marshmallow immediately, they get one. If they can wait for 15 minutes, they get two. And the kids who could wait did better in school and better in later life. And then Justice Ruth Bader Ginsburg told me in one of the interviews that I was honored to have with her that her mom, central advice to her was two things: first, be independent, and then be a lady. And by be a lady, she meant avoid unproductive emotions like anger, jealousy, and fear. They will distract you from productive work. And that classical idea that being a lady or a gentleman in the classical, not the aristocratic sense, meant self-possession, self-mastery, self-improvement, character improvement was the essence of the classical wisdom. So that is the understanding of the pursuit of happiness that prevailed for so much of American history. It made its way from the Declaration into the Constitution, where Madison's distinguished between reason and passion as the definition of faction and tries to set up a system to avoid the hasty formation of mobs. It's expressed in the Federalist Papers, which is a manual for public happiness, to use a phrase that Madison and Hamilton use repeatedly, and I hadn't noticed before. It's expressed by Tocqueville's idea of self-interest properly understood, where, channeling and reading Cicero, he also talks about the need for self-mastery and a virtuous citizenry. It ends up in books like the McGuffey Reader and the Columbian Orator, which inspired Frederick Douglass, who bought reading lessons on the streets of Baltimore with bread and then purchased this book with bread and read excerpts from Cicero and Franklin and Seneca and a dialogue on slavery, and turned himself into the greatest abolitionist of his time. And then it persisted in the early 20th century. My mom and Justice Ginsburg were born the same year and both went to New York public schools in Brooklyn, and they got it in their McGuffey readers and the textbooks of the time. And then it fell out of the curriculum in the 60s when the definition of happiness was transformed from being good to feeling good, from self-restraint to self-gratification. And that's a complicated story about why that happened, but it's obvious that once pop culture began to celebrate hedonism and materialism and not self-mastery and self-restraint, the definition of happiness changed. The happiness book tries to ask how the founders applied these lessons in their lives, and I'll just focus on how they applied it in their educational philosophy, because that's so central to the foundation of APS. Franklin, after having come up with his virtue project, founded a series of institutions in Philadelphia to inspire the cultivation of self-mastery and virtue. And APS was designed to ensure communication among, he called them the virtuosi, men, ingenious men from across the country and world who needed communications technology to be able to share their ideas, to cultivate information about the arts and sciences. But he also in 1728 had founded the Junto, the club to join, which was designed to inspire virtuous learning. It was a kind of reading group for his friends. He writes the Constitution from Locke's essay on education, which is designed for the promotion of virtue. Incidentally, the central definition of happiness, this is hiding in plain sight in Samuel Johnson's 1755 dictionary, two definitions: one, the Old English hap or luck; the second, the rational contraction of desire so that you can meet it. The footnote is to Locke's Essay Concerning Human Understanding, book 51, which defines the pursuit of happiness as the mastering of reason with passion. It was just literally hiding there in plain sight how the founders understood happiness, and Locke writes a manual for education and recommends that people get together and have self-improvement groups to do self-accounting of their efforts to achieve virtue. And that's what Franklin recommends. The Constitution asks questions, which he takes from Locke, without citation as it happens, asking what virtuous event have you described today? What vice? How can you promote virtue and avoid vice? And Franklin dreams of creating a united party for virtue, essentially a bunch of adult reading groups across the land that, like the APS, which he founds as a add-on to the Junto in the same spirit, will promote virtue. And then when he writes the first curriculum for the University of Pennsylvania, the main moral textbook that he recommends is by Samuel Johnson, not the British guy. They always get confused, but the moral philosopher, father of the Connecticut founding father at the convention. And you just do word searches because all these texts are online. He's constantly on the first page and throughout the volumes talking about the pursuit of happiness, which he defines as character improvement and virtuous self-mastery. And the whole curriculum is designed to inspire this in kids across the country. So that's Franklin. And isn't it amazing that at the end of his long life, he considered his greatest achievement, the great Franklin, the greatest man in the world, his greatest achievement is his tranquility of temperament, his conciliating temper that led him not to say tendentiously, my way or the highway, but it may be so, or perhaps, or it seems to me. That's what allows him at the Constitutional Convention to persuade people to make it unanimous, even though he knows that they have objections, and to be the greatest diplomat of his age. And then Jefferson, who, of course, falls short of so many of his ideals, most notoriously slavery, and there's much to say about that, including the fact that he perceived it, his own hypocrisy in classical terms, and recognized that it was his own avarice or greed, to use the classical terms, that led him to denounce slavery as immoral and at the same time to be unable to give up the lifestyle that it made possible because he liked it, and he was addicted to luxury and didn't want to free himself of that vice. But Jefferson puts on his tombstone the three achievements, not the presidency, although he no doubt prized his APS presidency highly, but writing the Declaration, writing the Virginia Declaration of Rights, and UVA. And his hope for democracy is the cultivation of a natural aristocracy, not a hereditary one. He has such a fierce enmity toward the inherited distinctions which subverted the ancient English liberties that he traced back to the mists of the Saxon woads. But he hopes that in this new democracy, universal education will inspire first the boys of best genius and then others to cultivate their faculties of reason so that universal reason can suffuse across the land. John Adams, in the most moving exchange in American letters, they're old and they've fallen out over politics. They get back together. What do they want to talk about? Comparative moral philosophy and education. Adams is so excited when he learns that Pythagoras, the founder of Greek moral philosophy, may have traveled to the East and read the Hindu Vedas, and he thinks if this can just be proven, it will show that the essential element of the East and West is similar. We are what we think. Life is shaped by the mind. Adams signals the passage in the Bhagavad Gita, renounce and enjoy, renounce attachment to external events and enjoy eternal salvation, and notes with his incredible deep reading that this is the same as the Stoic dichotomy of control, which says don't try to control the thoughts or emotions of others, only focus on controlling your own thoughts and emotions. And Jefferson says at the end of his long life, he's concluded he's no longer a Stoic, but an Epicurean, by which he means not a hedonist or wanton pleasure seeker. That's a libel on Epicurus, purpose, but the rational contraction of desires so they can be met. But they disagree about whether or not education is possible on a large scale. And Jefferson dreams of the perfectibility of man, and Adams scorns his starry-eyed faith in reason and says never since all of human history has man been perfected and we're fallen. And he's a follower of Polybius and thinks that all just aristocracies and oligarchies and democracies give way to their inferior forms of tyrannies and oligarchies and the mob. Both Jefferson and Adams, like all the founders, are centrally concerned about a Caesar that will subvert democracy, consolidate power, undermine the separation of powers, and that America will go the way of Greece and Rome. My new book, which is coming out in October, is about, it's called The Pursuit of Liberty, how Hamilton versus Jefferson ignited the lasting battle over power in America. And it begins with this incredible dinner party, not the one where they moved the capital from D.C. to the Potomac, the one in the musical, but a year later, they're in Philly, and at Washington's away, they're having a convivial working dinner. Hamilton looks around the room and says to Jefferson, "Who are those three guys on the wall?" "Those are my three visions of the greatest men in history, John Locke, Francis Bacon, and Isaac Newton." Hamilton pauses and says, "The greatest man in the world was Julius Caesar." And Jefferson writes, "This proves that Hamilton is for a monarchy bottomed on corruption," and then founds the Democratic Party in opposition to the supposed Caesarism and monarchism of Adams and the Federalists, leading to a polarization that will afflict American history until the end. Hamilton was almost certainly joking because he's always warning of the dangers of a Caesar who will court the people from below, flatter the mob, as in Shays' Rebellion, and install a dictatorship. Jefferson is concerned about a Caesar from above. He worries about a president in the distant future who will lose an election by a few votes, his exact words when he gets a copy of the Constitution, refuse to leave office, and install himself as a permanent dictator. They both, though, are dedicated to the separation of powers and committed to the possibility of civic education as a way of preserving the republic. And for the rest of American history, political parties and justices invoke Hamilton and Jefferson by name as they debate the great issues of national power versus states' rights, liberal versus strict construction of the Constitution, and democracy versus rule by elites. Friends, we're obviously living in a testing time where some fear that the founders' fears have come to pass and that Caesarism is replete in America. And a combination of the subversion of the separation of powers, which has been a long time in the making, including the vast explosion of executive power with a populist presidency, the shrinking of a Congress that is not performing its checking function, and a judiciary increasingly deferential to claims of a unitary executive, may, in the fears of some, be fulfilling the founders' fears. We don't know what the future will bring, but we do know about that the founders thought that the whole thing would collapse without virtue and civic education. And by that they meant individual self-mastery and taming our passions and not trying to own the libs or own the conservatives and being our most partisan and expressive selves, but being moderate and being self-composed and listening to the other side and disagreeing without being disagreeable. That beautiful scene, Hamilton dies, Jefferson places his bust in Monticello across from his own, and you can see it today, and when he would pass it, he would smile and say, opposed in life as in death. Not a hated enemy, but a respected enemy, respected adversary to be engaged. So what is to be done? I want to just say quickly, it's obvious that the age of social media is the antithesis of the calm moderation, temperance, prudence, and justice. And Madison and Jefferson had such faith in the new technologies of broadside press that would let citizens read the Federalist Papers in the original and talk about it with their representatives, and reason would slowly spread across the land. And of course, our world of Instagram and X and Enraged to Engage is in many ways the founders' media nightmare. But at the same time, there's a gleaming ray of hope. It is so exciting. We're so gloriously lucky to live in an age when all the books in the world are free and online. And it just blows my mind that I was able to do all of this marvelous reading just lying on my couch, reading these beautiful books, either in the original or free texts.
All we need is the self-discipline to read them. And that's why the greatest challenge to our civic life today is that people are not reading anymore. And not just the least privileged, the most privileged among us are not reading books. And we have it in our power to, I've become an evangelist for the transformative power of deep reading, and we have to model it ourselves. And for me, the biggest takeaway from that crazy COVID reading was I got back into the habit of reading every day. I'd fallen out of the habit. I read in college, and I read for my deadlines, but because I'm addicted to these screens, I'd stopped reading books. So my new rule is when I wake up, I'm not allowed to browse or surf until I read. And it's not always 2 hours before sunrise, but you know, it's just remarkable. It's so exciting I read John Newman on the idea of the university this morning, and just, you know, Rick Levin knows it's just, and that's really the Franklin classical faith too, about the possibility of daily cultivation and self-mastery and learning and growing for its own sake, because that's to unite with the divine. So back to APS, that is why your mission is so sacred, and your opportunity as a group of lifelong learners is so meaningful. How lucky you are to convene for a couple days and learn about disciplines different from your own for no other sake than for the joy of lifelong learning. This is the highest privilege that any of us can enjoy as citizens. I am so honored to be here. I'm so looking forward to our learning together, and let's take the light from this glorious learned society and inspire civic learning across America. Thank you so much. Great.
Time is upon us, but I think we have enough time for at least one question, or maybe two, if they're short. And please, someone bring a microphone and say your name and where you're from.
Ted Steinbach from Louisville, Kentucky, and I want to thank you for bringing to us the importance of liberal education with a small L. My question or comment to you is, here in Philadelphia, the physician humanist Sir William Osler spoke about equanimitas. Can you speak to that as one of the virtues?
Thank you so much. I think I was named Jeffrey for Jeffrey Osler, who was related to William Osler, and your Kentucky roots bring to mind one of my great heroes, Louis Brandeis, of course, who was such an Athenian and so devoted to this classical vision and considered 5th century Athens to be the apotheosis of the time of creating a perfect citizen in a perfect state. And equanimitas is exactly the definition of the first of the Aristotelian virtues. It's all about moderation, and it's temperance. And it's so interesting that both the classical and the Christian, both the later church fathers like Aquinas, who I think Osler maybe, I think equanimous may be from that, and the natural religion advocates like Wurlitzen and Tillotson all view a temperance as a synonym for virtue. And Adam Smith defines virtue as temperance because the roots are the same, and therefore equanimitas, like temperance, is itself a representation of the harmony of the souls. That's the extraordinary thing. You're trying to seek the same harmony in the constitution of the state as you seek in the constitution of your own mind. And only when reason, passion, and desire are aligned in that sense of a soul tranquilized by restraint and consistency, to use the Cicero language, can you achieve the equanimity that allows you to overcome your ego-based emotions and to connect to the divine, to the light within all, to the universal, sub specie eternitatis. It's the whole idea of the eternal soul, and you can only connect to it by achieving these virtues of temperament, and that's why it's a kind of temperance. If my Latin were better, I could show the roots of it all, but Smith did, and that's why equanimitas is a beautiful definition of the pursuit of happiness.
I love that. Wow. Thank you so much. I've got a few announcements before we adjourn. First, let's thank all of the speakers for just an incredible afternoon. So members of the council will be meeting now at 4 o'clock on the second floor of Philosophical Hall. Those of you who are not on the council, if you're interested in touring the library collections, please meet our director of the library, Michelle McDonald, in the foyer of Library Hall at 4:30. I think Michelle's up there. The cocktail reception will be here in Franklin Hall, beginning at 6 o'clock, so all this is going to be transformed. Those attending the new members' dinner, please make your way to Philosophical Hall at 7 o'clock after the reception. And we also have a special edition this evening from 8 to 10 p.m. There will be dessert and drinks available in the Copenhagen room on the ground floor of the Hotel Monaco. So this event is open to all meeting attendees, and you're welcome to drop by following whatever dinner plans you have. Breakfast will be here starting at 7:30 tomorrow, and the talks will resume at 9 o'clock. Again, my thanks to all of you, and thanks to the speakers. Thank you very much.